microvolumes, but it's not just about collecting small samples. What it is about is moving beyond conventional blood sampling processes and putting the patient at the center of the processes for collecting blood samples. At the moment, we're collecting blood samples where we're really thinking about the needs of the clinical trial, the clinical central lab, the bioanalytical lab, but we're not really putting the patient first in our considerations. So these technologies are really about how do we put the patient first. So what we're looking at is about collecting the appropriate sample. We want the sample to be good enough to answer the questions that we have. We want to use a process that's most convenient for the patient. We want to be able to provide high quality information. We don't want to downgrade the decision-making process, so we still want high quality data. And we want to be able to make high quality decisions. And this could be a small volume blood sample, or it could be a larger volume blood sample. As you can see from this slide, there are a number of approaches that are currently commercially available. We're not going to go into each of them, and we're not going to recommend any technology above the other, but all the technologies that are visible here are currently commercially available on the market. So what are the benefits of patient-centric blood sampling? Well, obviously we want to maintain the quality of our blood sampling. We want to obtain high quality blood, plasma, or serum sample for the accurate quantitative determination of our drugs, our drug metabolites, and endogenous molecules such as biomarkers, or for clinical chemistry. We want to put the benefits of the patient at the front. We want to minimize the impact on the human patient or the consumer. So we want to optimize the blood volume sample. We don't want to waste blood and throw it in the bin. We want to minimize the pain and invasiveness for the patient, and we want to maximize the convenience for the patient. We also have an opportunity here to enrich the data we generate. We want to still generate the high quality concentration data, but maybe we can do this in situations that are currently difficult or impossible to work with, such as the home or such as in certain medical events. So where might patient-centric sampling be of benefit? We really want to, as we're talking about, reduce the burden to the patient, particularly vulnerable patient populations. And today we're going to be talking about pediatric patient populations, but you can also imagine elderly, oncology patients, anemia, patients in rural locations. How do we include those into our clinical trials and our healthcare? We also have the opportunity to generate additional data for pharmacokinetics and for biomarkers. So this could lead to improved population PK modeling and an improved understanding of disease by having better exposure data or better data on our biomarkers. We also have an opportunity to generate data during a clinical event. For example, if you've got a migraine and we're wanting to find out, are you taking your medicine? Are you taking the right dose of your medicine? What's happening with your biomarkers? You're not really going to be able to get into a car and drive to a study center to have a blood sample drawn, but maybe you or a caregiver could collect a sample close to that migraine event that could give really unique data that just can't be obtained in any other way. We also think there's an opportunity here to improve clinical trial recruitment and retention. If people are able to collect samples in the comfort of their own home and don't have to go to a study center, then surely they'll be easier to recruit and maybe will be retained better on the clinical trial. There's also an opportunity to improve diversity by making this blood sampling easier. And we have an opportunity to better understand patient compliance. How often do you complete a clinical trial and wonder whether people really did take their medicine? We have an opportunity here to maybe sample our patients more often to just monitor whether they really are taking the medicine during the clinical trial. So with that done, I'm going to switch off my camera and I'll speak to you again later. Please submit your questions via the chat. But for now, I'm going to hand over to Enaksha to talk to you about small patients, big challenges. Take it away, Enaksha.
Thank you, Neil. Philip, can you give me control? Thank you. Thank you, everybody, and good morning, good afternoon. I'm so glad to be here to continue this conversation, and I'm going to be talking to you about small patients and the challenges that we face. Hopefully, everybody can hear me clearly.
Yes.
Okay, thank you. Oops, my slides are not. All right, so quick overview. I want to go through why do we need to even test drugs in children, right? And when we talk about children, who are the children and what ages, what's special about them? Talk a little bit about the legislation and regulatory requirements around conducting trials in pediatric populations, and then talk about the challenges from a parent's perspective, from enrollment, as well as from an economic perspective. Then I would like to touch briefly on the new buzzword that we are all excited about, decentralized clinical trials, talk about patient centricity and what that means, and then touch on blood sampling, because that's the core discussion that we are going to have today. And then how do we plan for tomorrow? How do we do these things better for tomorrow for our kids? So why do we need pediatric studies, right? Children get sick just like you and me, right? I mean, they all get sick and they all need medication. So there were statistics done in 2020, which stated that 25% of the global population is under 15 years of age. So that's 2 billion kids that would need medications if they are sick. But if you go back around the 2000s, or less than 25% of the drugs that were approved were labeled for pediatric use, right? So about 80% of the drugs were prescribed by doctors as off-label use. This number increased probably significantly, but not significantly enough. So in 2016, still less than 50% of the new drugs were labeled for pediatric use, and the remainder were obviously used off-label. So what do you mean by off-label? That's where the physician would adjust the adult dose, that's the approved dose, to match the child's weight and prescribe the appropriate milligrams or concentration of the drug. So what this does is when you use drugs off-label, it can put our children at risk, right? You can imagine if the dose is too high for the kid, you can end up with adverse events. If the dose is too low, there would not be any therapeutic effect, or there would be no benefit to the kids, right? And adverse events are reported as a common cause among all the hospitalized children. So a lot of the children that are hospitalized, among them, adverse events from drugs has been reported as a common cause. So as you can see, children can be labeled as therapeutic orphans, right? They don't have the regulatory or the approval that you and I as adults enjoy, right? So they need help, they need trials, they need medications that are approved for their use. So children are not small adults, right? As I said, you just can't take the child's weight divided by the adult's weight and prescribe medications, because many changes do happen in children or occur in children as they grow and develop, right? There's a lot of ADME properties that change as children grow, right? Especially related to drug clearance, right? The biotransformation pathways, elimination pathways, all of these mature with age and they change. The physiological changes happen in the GI tract that can affect absorption of your orally administered drugs. Changes happen in your body water, body fat content, right? These can have, and also there could be differences in protein binding that can impact the availability of your drug and the distribution. Drug metabolizing enzymes, right? Both phase 1 enzymes and phase 2 can change as children mature. It's well known and documented. For example, CYP3A4 increases with age, right? About 50% of the children reach about 50% of the adult activity by when they are a year old, and as they get older, would they reach the full activity that you and I have as adults, right? So So therefore, they would that would impact the metabolism and the availability of a CYP3A4 substrate drug. Renal clearance, renal function can change, and GFR reaches the value of adults only when the kid would reach about a year old, right? So different safety concerns, right? So as you can understand from what I just stated, the children may need a different dose. It could be higher or it could be lower, right? And here are 3 examples where the dosage that's administered to patients is actually higher compared to the adult dose. So therefore, drugs used in children must be studied in children, right? Studying them in adults is not gonna be a substitute. So let's take a look at who the pediatric patients are, right? This is challenging by itself because when you talk about children, it spans a broad range of ages from a preterm newborn infant all the way up to approximately 18 years, right? So you can see preterm newborns, term newborns are what we call neonates, infants and toddlers and children and adolescents, right? A broad range of growth stages of children, for which we need to make sure that the drugs that that are available have a specific and a prescribed dose for. The guidelines around the classification changes slightly compared to the ICH guidelines and FDA, and I have listed it there on the bottom. So what has happened, right? I mean, obviously our regulators, FDA and global regulators, do understand that there is a need for testing. And as in response to that, there has been several acts of passed to encourage the testing of drugs in children as well as to enforce these requirements, right? So 1997 was the Best Pharmaceutical for Children's Act, which is called as a BPCA. 2003 came the Pediatric Research Equity Act, which is called PREA or PREA. This was authorized in 2007 and made permanent in 2012. Then in 2016, the Race for Children's Act was passed, which basically required pediatric trials conducted even for pediatric oncology patients, right? So the BPCA was kind of a voluntary thing, and it was called the Pediatric Act, and the PREA allows enable FDA to require the conduct of pediatric studies. So this was more of a mandatory thing, right? Or the pediatric rule. So you can see kind of a carrot and stick scenario going on there. And what the FDA has also done, which most of you are probably familiar, is that now a pediatric study plan is mandated before you can submit an NDA, right? Failure to include a pediatric study plan with submission is a reason for refusal to file. So the FDA does mandate you to have a approved pediatric study plan for every new drug application. EMA also has a similar requirement. They call it a pediatric investigation plan or PIP. And this is also required at the time of submission. So the regulators are also mandating this or requiring drug manufacturers to think about conducting these trials in children. So since legislation was passed, about 50% of the drugs have been approved for pediatric use. So this is up from 25% from the data I just showed you. And overall, there has been about 300 pediatric label changes. So if you go back, remember the BPCA was a kind of a requirement, and the PREA was mandated. So you can see that the carrot gave about 48 drugs got approved, but with the stick, when the stick approach came into play, a large number of products have been actually studied and approved for pediatric use. So why are we talking about pediatric trials, right? Because pediatric trials are very difficult to conduct and enroll, right? So in 2016, there was a study done by Harvard University where they looked at 550 pediatric studies that were conducted between 2008 and 11, and this included 77,500 children. But the fact is that 40% of these studies never finished, right? They were terminated or they were finished and never published, right? So this questions the value of conducting these pediatric studies. And the biggest reason that was cited for trial failure was the inability to enroll. So the inability to enroll was the number one reason why these trials were terminated or failed, right? So Again, FDA and EMA has been very active in this area, trying to encourage people, helping people to conduct these studies. And one of there are two references here. I'm not going to discuss that, but just for a reference. But basically, encouraging people to have involve parents, caregivers, youth, children in these discussions, so that you can design these studies to be more favorable and attractive to the pediatric population. So what are the challenges for pediatric trials, right? The trial design itself can be quite challenging. It needs to be tailored to meet the needs of the children and their families. The dosage form can be challenging. We just can't give a big tablet to a little kid. So there's a lot of CMC related pharmacology activities that are needed to taste masking to liquid dosage forms, etc. But the ones that I think we need to kind of be very pay attention and concerned about is enrollment and consent, right? Because this involves emotions, right? Just imagine convincing a parent. I mean, most of you probably are parents, right? Would you like to enroll your kid in a clinical trial simply because somebody says that the experimental drug is better, right? You would also worry that whether maybe your child may get placed in the placebo group, right? Then what happens? Then also there are obviously sides of fears of side effects, right? Especially impacting a growing child's growth and development. And then comes the big things, right? Also trial procedures, right? The blood volumes, the multiple clinical visits, traveling out of state, taking time off, etc. So you can see that these do get very complicated from a personal, emotional level. And obviously there can be a lot of financial implications too, finding time and money, single parents, finding daycare for the other kids, time off from school, work, finding transportation, reimbursements, etc., right? So the take home is that this is not an easy thing, right? The running clinical trials pediatric clinical trials is a challenging task. So I mentioned blood volumes, right? So what are the challenges with collecting blood volumes? Since we're kind of gearing towards that discussion. Complicated dosing schedules, right? Timing of clinical visits, multiple clinical visits that you need to be at the clinic at a particular day at a particular time in order to ensure that your dosing is done right, and also collecting your blood samples, right? All the other trial related procedures. Multiple clinical visits, right? I mean, there's very rarely a clinical study where you come in on day one and then you're gone. You don't have to come back. There'll be 3, 4, 5, 6, maybe even a dozen clinical visits planned where you got to come back to the lab. And most of these visits will involve some kind of scans or multiple blood samples being collected. And these volumes of blood do add up, right? I mean, typically a Vacutainer tube would collect 2-3 milliliters of blood from a patient, right? For PK, we typically collect 2 milliliters of blood minimum for a PK time point, and we collect anywhere from 6 to 10 time points for a full PK profile. So this is the PK time points actually in addition to all the other standard safety labs that one would be drawing from a normal trial. So you can see that this can hinder recruitment and also can be a reason for dropout during the study. Why are there limitations in blood? Because as you can see from this reference and this table, there is a limit to the amount of blood that can be collected from a child, right? I'm not going to walk through this slide, but if you can look at a 12-month-old right on the middle there, you can only collect 24 milliliters of blood over a 4-week period, right? So this has to account for all the testing that you would do, including standard safety labs, which would be like your hematology and your chemistry panel and any other tests that are included, and all the blood draws that are done at screening, and all the PK draws that may be done on day 1, who knows, day 7, day 14, multiple times. So there isn't going to be enough blood to collect from these patients if we use traditional blood sampling schedules. So what are the risks of drawing large volumes of blood, right? I mean, these kids can have symptoms of anemia. Their hemoglobin levels can drop and would require additional volume replacement or iron supplementation, or maybe even blood transfusions, right, for really sick patients. It can be a very high risk for infants, right, the very smaller babies. And also multiple needle sticks or even accession to central lines or catheters can cause infections, right? There's a risk of infection. An additional caution for children who are associated with conditions related to inhibition of erythropoiesis, for example, bone marrow dysfunction or sepsis or anemia, or even patients undergoing chemotherapy, right? Which would be extra vulnerable to risks from blood draws. So we had some discussion some years ago, and this was statements that were made from Dr. Athena Zappa at Children's Hospital of Philadelphia. She basically said we need simplified approaches that avoid the use of central lines, right? Because obviously central lines can increase the risk of infection, minimize the blood sample volumes, as well as the pain that's incurred from a child. You can imagine the pain if you if you want to get multiple venipuncture from a child, and where appropriate, perform the sampling at an outpatient location, or maybe even some place that's more convenient to the patient. Could be even the patient's home. So there is a need to focus on the patient, right? So we need to, when we design trials, we need to think about the patient at the center, right? So how do we do that? We need to develop partnerships with the patients and the parents. We need to get their input, get them involved, just like the FDA and EMA recommendation. We have to think about their needs, right? We need to accommodate their needs, and also think about their convenience and engagement if we want to conduct successful trials and not have people or patients drop out or early terminate early from the trial. So this is something that we kind of did several years ago in 2018 at the International Children's Advisory Network Summit in Scotland. We were able to actually engage with pediatric patients, and we took one of these sampling kits and did a demonstration, gave them to kind of play around with it without sticking themselves, and ask the questions. I mean, would you want to collect a blood sample by yourself, as opposed to having to go to the doctor's office and have a needle stuck in your arm and get a vacutainer full of blood? And there was overwhelming acceptance or positiveness from these kids to use something like this, to do it themselves and do it away from home, right? So on the right hand side, I have some bullet points kind of capturing the responses. So we did a survey and the responses from the survey. But two key things that came out was the convenience and the privacy. I thought I was really blown away with when they talked about privacy, because they said now when my teachers or my friends ask me, why didn't you come to school yesterday? I don't have to tell them that I had to go to get a blood sample drawn, because I can do it at home by myself, right? So they like the fact that it's a smaller volume. They like the fact of being able to be responsible for their own care. And also, obviously not taking time away from their school or any of their extracurricular activities. So what did COVID do in the midst of all of this, right? I think COVID played a huge impact, and the two takeaways is that it basically accelerated and focused the need to decentralize patient or move towards patient centricity, because now we have an experienced audience, right? You, me, all of us. We are kind of used to doing things at home. We are kind of used to doing COVID testing at home. We are used to maybe even collecting a blood sample at home. I mean, people who may have been on certain COVID-related trials did collect blood samples at home using one of these, some sort of microsampling device, right? So we have a very captive, experienced audience. So, but microsampling has been, the uptake has been slow. By industry, right? So obviously it's not a panacea, right? It's not a one size fit all. It's also a disruptive technology, right? It's different to the status quo. There's always resistance to change, especially against the time-tested gold standard, which has been the vacutainer full of blood. Vacutainers were invented in late 1940s, early 1950s. So they've been around for 70 years now. And nobody wants to change that, right? But again, as I said, COVID did play a huge role in kind of moving that needle and making people more comfortable with alternative ways of doing things, including collecting blood samples or testing blood samples at home. So COVID did push the patient centricity over the hill. I think it's all downhill now. It's up to us to go forward, move these technologies forward, and see how we can use these technologies to help our patients and our clinical trials and develop drugs and also general healthcare. Right? I mean, these are things that can be applicable across the whole globe, so to speak. Challenges for microsampling, right? It has to be easy to use. It needs to be easy to collect. It has to generate data that's equivalent to plasma or the gold standard. It has to be scalable, right? Handle large numbers of samples. It has to be implementable globally, right? Across, if you're talking about clinical trials, multiple clinical sites globally. Some sites are resource limited. Some sites, are in very remote locations, so able to reach them. And these do have those advantages. Implementable at home. They got to be compatible with current workflows, and including automation. If you're running hundreds of thousands of samples, they need to be compatible with automation. And obviously, something has to be said about the cost, right? Savings, healthcare, cost or expense, right? Two ways to look at it. Again, kind of the same devices that were on Neil's slide, right? This is not not recommending any of these, but just to give a sense of the type of devices that are available. Some devices you can collect a sample of blood from a finger stick. Some devices you see on the bottom left, you can collect still a small volume of blood from your upper arm. So what is blood collected for and used for? There are numerous reasons and why we collect blood samples, right? From doing your basic chemistry or safety panels to other panels to disease markers, biomarkers, glucose testing, which people are already doing at home. Some specialty biomarkers, and obviously PK drug concentrations, right? Which is a big part of any clinical trial. To understand the pharmacokinetics of the drug that is being tested or being developed. So there has been a lot of guidance documents out very recently, and I just wanted to share some of them with you. They are all related to pediatric studies. The one on the left is from the ICH. The three on the next three ones are all from the FDA. Two of them are final, one is a draft, but they all do talk about conducting pediatric studies in guidance for conducting pediatric studies. So what do we need to do, right? I mean, we need to plan for tomorrow, right? Because the children's needs are urgent and they're real and they're global. So we need to be able to take bold steps. And so this is my last slide, and I would like to kind of pose the question, right? What's keeping you from adopting pediatric patients-friendly techniques and technologies? It doesn't have to be a finger stick, but we need to think more broadly and how we can bring these technologies to our clinical trials, and especially in this for this discussion, how do we help the conduct of pediatric trials by bringing these kind of technologies forward? So with that, I will be happy to answer any questions, I think, after the next talk. So let me stop sharing so the next talk can begin. Thank you.
Thank you, Enaksha. We're going to move over to Jeff now. So Jeff Silverman from Edgewise, over to you.
Thank you, Neil. So what I want to talk to you today is about my company's foray into patient-centric blood sampling. It's to enable some of our studies in patients with muscular dystrophies. And let's see if I can advance slides. No. So Neil.
Click on content, Jeff.
Content.
Yeah, at the top, if you click on content, you then be able to see next slide, or just click on your left and right arrow on your keyboard.
Got it. All right. So as I said, the primary objective of the study I'm going to talk about was to assess the PK of a compound known as Edgewise 5506. and we're investigating the use of a particular technology, which I'll go into in a minute, and evaluating that technology versus traditional venous blood sampling, as you just heard from Enaksha, the famous vacutainer. But the motivation for us to do this work was really because to facilitate collection of PK samples without requiring our patients to come into clinics and to reduce the number of traditional blood draws in a very sensitive patient population. And I'll go into that in a minute. So what I'm going to focus on is a study in adult healthy volunteers, so not a pediatric population, because this is where we were evaluating this technology and the data that we get from it. From the outset, I want to give a couple caveats. I am not an expert in microsampling or microdosing. I'm a clinical pharmacologist. I'm very used to using the traditional collection techniques. However, we're faced with this unique set of challenges because of our patient population. And so we needed to overcome this. So we started as a company to evaluate different technologies. We settled on the one that I'm going to talk about. But as you heard from both Neil and Enaksha, there are other technologies that are also viable options. So a little bit about 5506, which will enable you to understand the context from which we're coming. It's a novel, orally bioavailable small molecule that's used to modulate type 2 skeletal myosin. And our goal is to protect dystrophic muscles from contraction-induced damage. And by protecting those fast muscle fibers, 5506, we're hoping, where this drug is in early clinical studies, we're in phase 2. We're hoping that it limits muscle breakdown and disease progression, particularly in Duchenne's muscular dystrophy, but we're also investigating in other forms of dystrophinopathies like limb-girdle and Becker's muscular dystrophy. As I said, this compound is currently in phase 2. So the overall study design that what I'm going to talk about is it's an open label, 1 period, single dose study in which we administered 5506 to adult healthy volunteers. We collected blood and plasma in several different ways. We collected venous blood, so we had whole blood, converted part of that to plasma, so we had a plasma sample. And then the last one is we collected using a device called a Tasso M20. As I said, some of the advantages are, and Neil and Enaksha both said this, it allows for collection, the Tasso device allows for collection of samples at home. It also, the side benefit of reducing pain and stress, we're in this patient population when in the clinical studies in young boys with muscular dystrophy. Some of these boys are very, very sensitive or adverse to traditional needle sticks, and we've had a number of patients refuse to enter a clinical trial because of that fear. And so switching to this alternate way of collecting the samples may allow those boys to overcome their fear and allow them to participate in the trials. It also allows us to collect samples throughout the study. We're not requiring people to come in on every visit to a clinic. Some of our patients live a long distance from the clinical site in which the trial is being held. So it allows us to collect samples, and it's a huge convenience. It allows us to collect samples throughout the day, different times of the day. And obviously we can send phlebotomists out to the home, but between the fear and the cost and the logistics of that, collecting with this Tasso device overcomes many of those challenges. So what is this Tasso device? Here I took this from the Tasso website, and you can see a picture of it in the middle here. It's really a very simple device. It gets attached to the upper arm, push a button, and it collects collects 17.5 microliters in 4 different samples, and then it's a dried blood spot in the end of the day. And so we needed to investigate whether that dried blood spot technology was amenable for our compound 5506. So as I said, the primary focus for us was comparing traditional plasma collection to this capillary blood collection. Although, as I alluded to, we're also comparing plasma to whole blood using venipuncture and venipuncture or whole blood venipuncture to capillary device. So there's 3 different types of samples. Very high level, if we look at plasma concentration time curves, you can see that all 3 methods give us roughly comparable data. That's at a very high level, very satisfying. It gave us a lot of hope that we could use the Tasso device in this clinical trial and in our future trials in patients. When we look a little more carefully at the variability between the methods, the one that we're focused on is here, M1 versus M3. And that's capillary sampling versus venous plasma sampling. And you can see that the level of variability, so on the y-axis is percentage difference. 0 would be perfect, right? And so you can see that we're not at 0 and there's a high level of variability. And when we look at other types of samples, so capillary versus whole blood, there's even higher variability. So we needed to understand that a little better and to know how comparable were these methods. So here you can see, again, it's a slightly different way of looking at it. It's the percentage difference on the y-axis and time on the x-axis, and this is for whole blood or plasma versus Tasso device. And what we observed very clearly here is the high variability really occurred in the first time period. I'll call it the first 6 to 12 hours, after which there was almost complete agreement between plasma and Tasso. And so we needed to understand a little bit more about this variability, whether it's due to the technology or due to physiology. And another way of looking at this is called the Bland-Altman. Some of you may know this analysis as a Tukey mean difference plot. So when it's a way of looking at 2 different measurements and seeing how much they agree. When they agree, that's called identity. So the line of identity is the blue line. That's and then it's flat. The 2 standard deviation lines are the red dash. 3 standard deviations are the green dash. And so we're looking at on the y-axis the difference versus the average concentrations, and you can see that the vast majority of the good concordant data are here circled in red, and those are at the later time points, the lower concentrations. The earlier points at the higher concentrations are are to the right on this graph, and you can see that's when they get a little discordant. But the vast majority of them are within the 2 standard deviations. And if you're within 2 standard deviations with this, actually 1.96, but call it 2 standard deviations on this Bland-Altman analysis, that's considered that the 2 methods are used interchangeably or can be used interchangeably, assuming 2 standard deviation is not a clinically significant difference in concentrations, and for this drug it's not. So this is giving us at least a first look, early look of that these sample collection techniques could be used interchangeably. So between plasma and capillary sampling using the TASO, we're feeling that the concentrations that we obtained are similar enough to be able to use the device, and that the early time point differences we think are probably more representative of pharmacokinetic distribution, capillary blood versus plasma. So the capillary is sensing a higher degree of variability of the sample based on other studies as well. And so for our intended use, where these boys are going to come into the clinic for their initial evaluation and enrollment on the trial, and then they go home and continue on their drug. So they're at later time points. They'll be at steady state for the drug. So we'll be out really on this part of the plasma curve, the right-hand part of the plasma concentration curve. We think that the TASO device is a reasonably good option to use. So we did obviously some other comparisons, venous blood versus venous plasma. And you can see here, we're looking at the percent difference. And again, the early time points have high variability. A little more difference at the later time points compared to the other, the TASO versus the plasma. But Bland-Altman analysis, again, the blue line is identity. The red line is the mean difference. And those are very tight to each other. And you can see a vast majority of the samples are within the 2 standard deviations. Again, suggesting that these 2 methods will be able to use interchangeably for the data. The last comparison that we did was capillary blood versus venous blood. And you can see here the difference curve out at the later time points. The difference is going away. It's much smaller than the early time points. And then finally the Bland-Altman analysis, again, the identity line is very close to the mean difference. And the majority of the samples, particularly at the lower concentrations, so later time points, are within the 2 standard deviations. So we really believe that the difference between the capillaries, it's a difference between capillary sampling and venous sampling is the most important, and that difference is largely reflective of pharmacokinetic distribution kinetics rather than the technical manner in which the samples were collected. So a high-level summary from a science perspective is the drug levels from all 3 of these collection methods are generally consistent, and that'll facilitate sample collection in our patient trials. The early time point differences between capillary and plasma sampling, traditional vacutainer, were really due to the distribution kinetics. And the use of this TASO device will certainly help us with sample collection on days where clinic visits aren't scheduled or possible, and it will reduce the fear factor in the young boys, and it will allow us to collect samples from those patients who are very, very reluctant to have traditional venipuncture. All I've talked about today is pharmacokinetic sampling. We are also exploring the use of the same device for pharmacodynamic samples. We measure a number of biomarkers throughout our studies, and we're currently validating the use of the TASO with each of those different pharmacodynamic measurements. And most important for this audience and today is, what has our trial experience been to date in a Duchenne's population? So now this is in young boys. I think the youngest on the trial so far is a little under 4 years old, and their patient and caregiver experience. The families said it's cool. They like it. It's easy to use. They really appreciate not having to travel to the clinical site. As I said, sometimes our patients, it's an airplane ride, not just a drive to get to the clinical site. So the families, they don't have to take off from work, skip school, and it's much easier to get additional blood draws from these small patients. The patient or the parents usually like that the samples are shipped at room temperature. So this device gets attached to the arm for approximately 5 minutes. After it's taken off, it's put in an envelope and shipped back to our clinical site in normal postal service. We don't need to have it stored on dry ice or wet ice. It doesn't have to be shipped by a expedited courier like UPS or FedEx. And the patients really liked being able to just ship by a postal service. So far, we only have a limited number of people in this Duchenne's trial, but those that have used the device really feel that it went very smoothly. We've had very few hiccups with it. We have had some trouble in a few cases where they've had trouble getting the sampling device to fully fill. We provide a backup device and give advice on counseling on how to use the device a little more. Either heat or somehow warm the site of collection often works, and then the collection goes very smoothly. So, in summary, this is Edgewise's first foray into using this type of patient-centric sampling collection technique. I would say it's been a success. We'll be able to get good pharmacokinetic data, and the patients are having a much easier way of collecting those samples for us. So with that, thank you for your attention, and I'll turn it back over to Neil and for questions and answers.
Thank you, Jeff. That was a fantastic presentation. Thank you so much. Before we go to the panel discussion, I'm just aware that we're getting close to the top of the hour, so I just want to make a couple of quick plugs before we move into the Q&As. Just for those of you that are not aware, there's a few organizations out there that are trying to work across organizational boundaries to help promote the understanding and use of these technologies where they are appropriate. One of these is the Patient-Centric Sampling Interest Group, which has been around a little while. We're currently turning ourselves into a not-for-profit organization, and we have a number of activities ongoing around education, around surveys. We're running a clinical trial. We have a very active diagnostics working group, and we have a community on LinkedIn. If people are interested in joining this group, please just look up the group on LinkedIn or write to me and we can get you involved, and you'll be hearing a lot more about the group in the future. There's also, for those of you who are in the pharmaceutical industry or contract research organization, There's an AAPS subgroup called the Microsampling and Patient-Centric subgroup, which is part of the AAPS bioanalytical community. And again, they've got a lot of activities going on with webinars, education pieces, and organizing sessions at different events. Again, if you're interested in being involved in this group, Enaksha is one of the co-chairs and Shefali is the other co-chair. Their email addresses are there. Please contact them. These slides will be available on the evidence base after this event. So you'll be able to review this and get these contacts and links to some of the different technologies that are available through those slides that will be permanently available on the evidence base. Just before we go to the discussion, I'd just like to thank our sponsors again, who we could not have done this without them, and thank the evidence base and Phil for giving us this platform for working with you today. So I want to go back to the panel discussion and some of the questions that have come in. So one of the questions we've got here is, do these techniques work for all drugs? And does anyone want to take that question at all?
Yeah, Neil, I can take a stab at it. I think initially most of the work with microsampling was basically limited to small molecule drugs, but I think over the past years there has been numerous success stories broadening the adoption of these kind of technologies to monoclonal antibodies as well as other novel therapeutics, right? So I think it's clearly it's doable. It's just a matter of, it may require a little additional method development, but there are it's definitely doable.
Yeah, and even within small molecules, so the real question is, how stable is your drug, and is it amenable to dried blood sampling? You know, some drugs are, some drugs aren't. I'd say the vast majority are, but that's one consideration. And to Enaksha's point, we're using this now for, or beginning to move into biomarkers, which again is large molecules. So.
So some other questions, there's a lot of questions coming in, and I don't think we're going to get time to cover them all within this event. So I'm hopeful we'll be able to answer some of them and post it onto the webpage after the event. So I apologize if your question doesn't get answered in this time, and some of them are quite complicated as well. Just another question that's come in is, what is the maximum frequency of sample collection from the upper arm, please? Anyone got any data on that?
Maximum volume?
Maximum frequency of sample collection.
Frequency. We actually haven't tested that. You know, in my own experience, which is very limited, we've only collected at hourly intervals, or even much less frequent. You know, in the Duchenne's, we only do it once a day, so it's sparse sampling. So
yeah, I think there's been some, I can't remember the exact number, and maybe we can look this up afterwards. I think there's been a Merck publication that goes into this a little bit, and I think they stated between 6 and 8 samples per day from the upper arm, 3 or 4 on each arm. And actually, do you have any insight into that at all?
No, I think I kind of recall what you said too. I was gonna just guesstimate like 6 to 8. You know, I read or heard that it's possible people have collected a full profile. Obviously, you have both arms, so you can do 3 on one, 3 on the other within a 24-hour period. I think I think the manufacturers might have more information on that.
Yeah, but certainly if you're wanting to get time points close together, you may have difficulties. And I think that's reflected in the data that Jeff was showing, where at the kind of later time points at steady state, you were getting higher quality data than the earlier time points.
So, Neil, if I may just take a minute and add. So one thing I think we need to all remember is that this is not a one size fits all. It's not going to work in all situations, right? This is a technique that's going to enable us to do things that were either not possible or we failed at before, right? So it's to get that additional time point, it's to get that important time point, it's to get that small volume of blood from a neonate, right? So we need to, I think, be a little bit more focused in our thinking and not think that, okay, I can put this in my next trial and expect it to work perfectly for everything else after that, right? I think each one of these, and you see that there are different types of devices and they may be suitable for different applications too. So there are choices. I think there are a lot of, I call it a lot of tools in the toolbox. You got to use the right tool for the right application.
Yeah, absolutely. Maybe we have time for one more question. If I just scroll back up through the questions. Any insight on IRB feedback and approval of a patient-centric approach in pediatric trials? So when you went to the IRB with these kind of studies, what kind of feedback was obtained?
For pediatric trials, we have run pediatric trials and we have not had any pushback. So obviously questions do get asked, is this device FDA approved? Is the device CE marked? Is it available? If it's a global trial, is it available in the countries that you're running the trial? So those kind of questions do come up, but nothing that we have not been able to respond to and get a favorable or move forward with.
Okay, thank you, Enaksha. So I think we've gone past the top of the hour and we've passed the hour for this event. The Patient-Centric Sampling Interest Group will be bringing you more webinars and more events at conferences in the coming months. So please watch your inbox and the website and the LinkedIn group for more events in the future. I'd like to bring this event to a close. I'd like to thank Enaksha and Jeff for their wonderful presentations. And I'd like to thank Phil and the Evidence Base for giving us the platform to be able to bring this to you today. And I'd like to thank all the attendees for spending their time today. Unfortunately, we didn't have time to answer all your questions. We'll make an effort, if we can capture these questions, to answer them after the event. And if we don't do that adequately, hopefully you can bring them to an event coming to a laptop near you in the not too distant future. So thank you all and enjoy the rest of your day.
Thanks. Bye-bye.